allow materials to get into the cell, and it cannot be able to allow materials to get out of the cell. The last property of the cell membrane is it is polarized. Polarized means that it has electric charges. The outer part of the cell membrane has positive charges, while the inner part of the cell membrane has negative charges. Now, all these charges enable that particular cell membrane to detect changes in its environment. I hope you're taking notes on these main points. Okay, we're going to draw a structure. We're going to draw a structure of the cell membrane so that we can have a visualization of what we are talking about. Structure of the cell membrane, so that we can be able to visualize what we are talking about here. So a cell membrane is made up of a phospholipid layer. It's made up of a phospholipid layer. And the cell membrane also contains spaces, and those spaces are called pores. So it is in these spaces that exchange of materials takes place. It is in these very spaces or pores that exchange of materials takes place. Now, what surrounds the spaces are proteins. So this is a protein, and this on this other side is a protein. So the space that is left here is what we are calling the pore. And it is through this space materials will exit the cell, and it is also through this space that materials will get into the cell. Now, this all together is what we are calling phospholipid. This is what we are calling phospholipid. And as we have said, the cell membrane has electric charges. So this is the outer part of the cell membrane, which has positive charges, and the inner part has negative charges. That is the structure of the cell membrane. With that review, we can now be able to get into our main focus for today. Because we are learning about cell physiology, we have processes that enable materials to get into the cell. We have processes that enable materials to get into the cell. And we say some of these materials might be gases, might be water, maybe some substances such as glucose. So a physiological process is one that allows materials to get into the cell. We have three main types of physiological processes. And the first one is diffusion. The second physiological process is osmosis. And the third physiological process is active transport. So these three allow materials to get in and out of the cell. But today, our main focus will be diffusion. Our main focus will be diffusion. And in diffusion, these are the key areas we are going to look at. Number one, we are going to define the term diffusion. What does it mean? We are also going to define other terms related to diffusion. For example, the term diffusion gradient. We are also going to look at experiments that are used to demonstrate diffusion. And we are going to look at factors affecting diffusion. What makes diffusion to be high? What makes diffusion to be low? Last but not least, we are going to look at the role of diffusion in living organisms. What is the function of diffusion? What does it serve in the bodies of living organisms? With me here, I have a snow spray. With me here, I have a snow spray. And this snow spray contains a lot of snow spray inside. This is a container. And this container is housing the snow spray inside. So the amount of snow spray inside here is a lot. The environment we are in right now does not have snow spray, but the snow spray inside here is a lot. So I can say that the concentration of snow spray inside this container is greater than that of the environment. The concentration of snow spray inside this container is greater than that of the environment. So what I'm going to do, I'm going to spray, and let's observe what will happen. I'm going to spray. So I have sprayed. So what has happened, the snow has moved from its region of high concentration to another region of low concentration. The snow has moved from this container where it is in great amounts to another area where it is in less amount. And this is simply diffusion. And so, diffusion is the movement of particles from a region of high concentration. Diffusion is the movement of So it's the movement of particles. Let me use a darker pen. So it is the movement of particles. Movement of particles from a region of high concentration to a region of low concentration Yes, that is the definition of diffusion. That these particles, the molecules, will move from where they are in large amounts to where they are in little amounts. So this brings us to the second concept we are going to define, and that is diffusion gradient. Diffusion gradient. Diffusion gradient. From this definition, we find that we have two regions. The region of high concentration and the region of low concentration. When we talk about concentration, we are talking about the amount. So the amount of particles in one region is high, and the amount of particles in another region is low. There is a difference between the amount in the first region, which is high, and the amount in the second region, which is low. So diffusion gradient is the difference in concentration of particles between the region of high concentration and the region of low concentration. We can write that down, that it is the difference. It is the difference. It is the difference in concentration of particles between region of high concentration concentration and region of low concentration. Region of low concentration. We're going to demonstrate that using our board. If this is our region of high concentration, high, and we have our region of low concentration. So we are going to assume that the particles of snow in this region were 100, and they spread to their region of low concentration. So let's say the ones that spread were 20. So diffusion gradient is simply 100 minus 20. That is 80. This is what we're calling diffusion gradient. It is the difference in the particle concentration. The high region minus the low region, you'll get that difference. The word diffusion gradient can also be called concentration gradient. Diffusion gradient can also be called concentration gradient. We are done with the first part of our discussion, that is definition of terms. Now we are going to focus on the second part of our discussion, which is experiments that demonstrate diffusion. Experiments that demonstrate diffusion. The first experiment involves a chemical or a compound that is called potassium manganate 7. So potassium manganate 7 is purple in color, and how it is used to demonstrate diffusion, a crystal of potassium manganate 7 is put in a glass rod. This is a glass rod. This is a beaker, an empty beaker. So a crystal of potassium manganate 7 will be put inside the glass rod until it falls to the lower region. So this is the crystal. So after that, water will be added into the beaker. Water will be added into the beaker. This is water. It is added into the empty beaker. Then the glass rod is removed. The glass rod is removed so that this particle remains at the base. And so what will happen, the particles of this crystal will break away. They will break away and they will start spreading inside the water. So this is the crystal on the lower side. So this is the particle of potassium manganate 7, and the particles that compose it will break away. Breaking away means that they will spread themselves, and after a while, all the water will turn purple in color. After a while, all the water will turn purple in color. So this is water. Then the dots I'm going to draw, the circular dots, are the ones that are presenting the particles of potassium manganate 7. They are going to spread evenly in the water. The observation, what is seen after this experiment, is that the water turned purple. The water turned purple. The explanation for this observation is the water turned purple because the particles that are making potassium manganate 7 broke away from the crystal and spread throughout the water, and they spread through the process of diffusion. What made these particles spread is the process of diffusion. So you can see that every molecule, every particle is moving from where it is in high concentration, that is this point, and it is spreading to where it is in low concentration. That is the first experiment that is used to demonstrate diffusion. The second experiment that is used to demonstrate diffusion is use of iodine and starch. In the laboratory, we do not have live cell membranes. We cannot remove cell membranes from live organisms and bring them to the lab. So what we have in the lab to represent the cell membrane is called a Viskin tubing. What we have in the lab to represent a cell membrane is called a Viskin tubing. A Viskin tubing is a clear paper that takes this shape, and this clear paper is semi-permeable. It is semi-permeable, meaning that it can only allow some materials to get inside it, and it doesn't allow others to pass through it. The second experiment will involve the use of the Viskin tubing. So this is the Viskin tubing. And we have said the Viskin tubing represents the cell membrane in biology experiments done in the laboratory. Now we have a beaker here, and in this beaker we have a solution, iodine. So iodine is brown in color. Iodine is a solution or a reagent that is used in the lab. It is a dye, and that is why it is brown in color. It is a dye, and that is why it is brown in color. Then in this experiment, what will be put in the Viskin tubing is a starch solution. Starch solution. Starch is a compound. It's an example of a complex carbohydrate. So when you mix starch with water, you get starch solution. Starch solution is white in color. Starch solution is white in color. Now, when iodine reacts with starch, it forms a blue-black solution. When iodine, this chemical in the beaker, reacts with starch, it forms a blue-black solution. And so, in the lab, we say iodine is used to test for starch. This starch will be kept in the Viskin tubing. So this is starch. It will be kept in the Viskin tubing, and the Viskin tubing will be closed at both ends to ensure there is no leakage. So what is inside here is the starch solution. Starch solution. So on one side, a string will be tied on one side, and this starch solution will be lowered in the iodine. This starch solution now will be lowered here. And the experiment will be left to stand for 20 minutes. The experiment will be left to stand for 20 minutes. The experiment will be left to stand for 20 minutes. So at the beginning, this is the beginning of the experiment. This is the beginning of the experiment. The starch solution will be white in color. It will be white in color. But after 20 minutes, that is at the end of the experiment, the starch solution will have turned from white and it will have formed a blue-black solution. So this is the same beaker, and this is the start solution, and it will have formed a blue-black solution. It will have formed a blue-black solution. The observation after this starch solution is removed is that it will have turned from white to blue-black. An observation in biology is what you can see. What can you see? If you're given this experiment and if you do it practically in the laboratory, what are you seeing? That is the observation. So the observation will be the start solution will turn from white to blue-black. And this means that iodine is in high concentration inside the beaker. Iodine is in high concentration inside the beaker. So the molecules of iodine will diffuse into the starch solution, and this iodine will react with the starch to form a blue-black solution. The iodine molecules are greater in the beaker, so they will move from their region of high concentration, which is in the beaker, inside the viscous tubing where the starch solution is. That is, the starch solution is their region of low concentration. So when they move inside through the process of diffusion, this starch will react with the iodine to form a blue-black solution. So that is the second experiment that is used to demonstrate diffusion in the lab. I hope we are together. You can send your questions if you have any. You can send your questions through the phone number that is below your screen so that we can have an interactive session. So we are going to continue. After the experiments, we are going to look at what affects diffusion. What makes diffusion to be fast? What makes diffusion to be slow? What makes diffusion to be higher? And what makes it to be low? So there are factors that affect the rate of diffusion. Rate means how fast or how slow something happens. So there are factors that affect how fast diffusion takes place or how slow diffusion takes place. Now these factors are 7 in number, and there's an acronym that you can use to remember these factors. Factors that affect diffusion. There are 7 in number. There are 7 in number. So there's an acronym that you can use to remember these 7. So you write D, T cubed, S cubed. D, T cubed, S cubed. So D stands for diffusion gradient. D stands for diffusion gradient. Then T cubed means that T has to appear 3 times. So the first T is temperature. The second T is type of medium. The third T is thickness of membrane. Thickness of membrane. Then we have S cubed. S cubed means that S has to appear 3 times. So the first S is size of molecules. Size of molecules. The second S is surface area, and the last S is surface area to volume ratio. Surface area to volume ratio. Surface area to volume ratio. So these are the 7 factors that affect the rate of diffusion. So we can state them first, then we are going to explain each of them. Diffusion gradient, also called concentration gradient, temperature, type of medium, thickness of membrane, size of molecules, surface area, and lastly, surface area to volume ratio. We're going to start with diffusion gradient. As I had defined before, diffusion gradient is the difference in particles between the region of high concentration and the region of low concentration. So when the difference is high, the rate of diffusion is high. And when the difference is low, the rate of diffusion will be low. We can demonstrate that. We had this small table here showing how particles are in the region of high concentration and in the region of low concentration. So in the region of high concentration, we're going to have 2 scenarios. This is the first one. In the region of high, there are 100 particles. When in the region of low, we have 20 particles. The difference or the diffusion gradient will be 100 minus 20, which is 80. Then we have another scenario where we have 100 in the region of high concentration and 2 in the region of low concentration. The diffusion gradient will be 100 minus 2, which is 98. So diffusion gradient here on this second scenario or in the second scenario will be greater than in the first scenario. So there will be a higher rate of diffusion in this area because the difference in concentration is greater, and there will be a low rate of diffusion in this area because the difference is lesser. So the higher the concentration gradient, the greater the rate of diffusion. The lower the concentration gradient, the lower the rate of diffusion. So don't get confused. Concentration gradient and diffusion gradient mean the same thing. After that, we have temperature. So temperature increases the energy content of the diffusing molecules. Increase in temperature increases the rate at which these molecules will move. So increase in temperature increases the rate of diffusion, while decrease in temperature decreases the rate of diffusion. Then we have another factor, that is the type of medium. A medium is where something is taking place, or it can be a reaction, it can be a movement. So we have 3 types of mediums. That is the 3 states of matter: solids, liquids, and gases. Diffusion does not take place in solids. Diffusion does not take place in solids, but it takes place in gases and liquids. The thing is, diffusion is faster in gases than in liquids. When particles are diffusing, for example in gases, gases can be this open environment because the atmosphere is made up of many gases. So when particles are moving around this area, they will move faster compared to the same particles moving in water. So diffusion is faster in gases than in liquids. The other one is thickness of membrane. thickness of membrane. Now a membrane, for example, a cell membrane, as we looked at when we were beginning, can either be thick or thin. So we're going to draw two membranes. The first one will be a thick membrane. Thick means that the space in between the both sides is great. Now this is a thick membrane. This is thick. And we have another membrane that is thin. Thin, like this. It is thin. So in thick membranes such as this, the rate of diffusion is slow. In thick membranes such as this, the rate of diffusion is slow. The reason why the rate of diffusion is slow is because the diffusing molecules will travel a longer distance. This distance is long, so they will travel a longer distance to the other side to cross to their region of low concentration. So this makes the rate of diffusion to be slow. Then we have this membrane. As you can see, the size between the length between the far side and the lower side is less. So the diffusing molecules will only take a short time to reach the next region. So in thin membranes, the rate of diffusion is fast. In thin membranes, this is a thin membrane, the rate of diffusion is fast. The other factor that affects diffusion is the size of molecules. Molecules are also called particles. Molecules are also called particles. Molecules are also called particles. Now we have two categories of molecules or particles. We have large and heavy molecules. Large and heavy molecules. And we have small and light molecules. Small and light molecules. So large and heavy molecules, we can draw big shaded circles to demonstrate this. Large and heavy. They are big in size and their weight is more. Then we have these two unshaded circles to present small and light molecules. So small and light molecules, they diffuse faster than heavy and large molecules. Small and light molecules will diffuse faster. They will diffuse faster than heavy and large molecules. Heavy and large molecules will diffuse slowly due to their size and due to their weight. The other factor that affects diffusion is the surface area. Surface area. What amount of area is exposed for diffusion to take place? We can demonstrate that by drawing two surfaces. We have surface A and surface B. This is surface A and this is surface B. So the first surface is big in size and the second surface is small in size. So the rate of diffusion will be high where the surface exposed is large, because a lot of molecules will be able to get to their region of low concentration. But where the surface area exposed is small, the rate of diffusion will be low. This is because a lesser area has been provided for diffusion to take place. So this is big, meaning that more molecules will diffuse, but this on other side, this area, surface B, is small. meaning only a few molecules or particles can move to their region of low concentration. Last but not least, we have surface area to volume ratio. Now, surface area to volume ratio is a comparison between the surface exposed, for example, for an organism, versus its volume. So the higher the surface area to volume ratio, the higher the rate of diffusion. The higher the surface area to volume ratio, the higher the rate of diffusion. So those are the 7 factors that affect the rate of diffusion. Students, when you're outlining these factors and explaining them, you must give the both sides. For example, if it's temperature, you must say, how does high temperature affect diffusion? Then explain also how low temperature affects diffusion. Don't leave your notes hanging. Don't leave your answers hanging. Now, from these points, we are going to look at the role of diffusion. Role of diffusion. When I talk about role, I'm talking about the function. What is the function of diffusion? How does it affect? How does it help living organisms? This is the role of diffusion, also known as the function. The importance of diffusion. Role of diffusion. So we have these major roles of diffusion. Number 1, diffusion helps in absorption of materials in plants. It helps in absorption of mineral ions in plants. Diffusion also helps in absorption of end products of digestion in the small intestines, and products of digestion in the small intestines. It also aids or helps in gaseous exchange in plants. Gaseous exchange in plants. It assists in gaseous exchange in animals. Gaseous exchange in animals. Gaseous exchange in animals. Diffusion also helps in excretion of nitrogenous waste in unicellular organisms. Excretion of nitrogenous wastes in unicellular organisms. Unicellular organisms. And lastly, it helps in translocation. It helps in translocation. Location of end products of photosynthesis. End products of photosynthesis. End products of photosynthesis. These are the main importances or the major roles of diffusion in living organisms. This diffusion process assists both plants and animals. For example, it helps in absorption of mineral ions in plants. Now, mineral ions are found in the soil solution. Soil water is called soil solution. So these mineral ions are in higher concentration inside the soil solution. So what happens? They will diffuse from the soil solution, which is their region of high concentration, and they will enter into the root hairs where they are lowly concentrated. These mineral ions will then be taken up by the plant and used in the body. The second one is absorption of end products of digestion in the small intestines. Living organisms such as mammals, we be specific on human beings, the process of digestion ends in the small intestine. Digestion is the breakdown of complex food into simple substances. The simple substances formed include glucose. So for glucose to be transported into the body, it first has to diffuse from the small intestines into the bloodstream. Then it is then transported to all cells where it is used for processes such as respiration. Another function of diffusion is that it aids in gaseous exchange in plants. Diffusion aids in gaseous exchange in plants. Gaseous exchange is the process where respiratory gases, oxygen, and carbon dioxide pass across respiratory surfaces. Now, gaseous exchange in plants, during the day, the plants take in carbon dioxide gas, which they use for the process of photosynthesis. After photosynthesis has taken place, this plant gives out oxygen as a byproduct. The process that the plants will get the carbon dioxide and the process that the plant will remove the oxygen after photosynthesis is through diffusion. Now, this process is a reverse during the night. In the night, the plants take in oxygen and they also give out carbon dioxide through the process of diffusion. Next, we have gaseous exchange in animals. In gaseous exchange, we be specific on human beings. Gaseous exchange takes place in the air sacs. So when you breathe in, that air is rich in oxygen. So oxygen will diffuse from the air sacs into the bloodstream, and carbon dioxide, which is a waste gas, will diffuse from the bloodstream into the air sacs, and finally you will breathe it out so that it can be excreted to the external environment. We also have excretion of nitrogenous waste in unicellular organisms. Nitrogenous wastes are wastes that contain nitrogen as a component. Unicellular organisms are those that are made up of one cell only. For example, amoeba. So in amoeba, what will happen? This nitrogenous waste is greater in concentration inside the body of the amoeba, assuming this is amoeba. So if this is our nitrogenous waste, it is in greater concentration inside the body, and now it will get out of the body of that amoeba through the process of diffusion. Lastly, we have translocation of end products of photosynthesis. Translocation is the process that end products of photosynthesis, such as glucose, are removed from the leaves and are transported to the rest of the plant. Now, because the leaves are the major points where photosynthesis takes place, this glucose cannot be retained here. It has to be transported to other parts of the plant. So what aids this transportation or this translocation is the process of diffusion. So that is it with the role of diffusion in living organisms. And with that, you can be able to answer any question in diffusion. So I'm going to leave you an assignment. You can tackle this assignment and it will help gauge how much you've understood from this topic. A very simple topic which has had testing even in the main exam. So the following questions I'm going to write on the board are from the main exam. These questions can be brought as short questions, structured questions, and even essay questions. So what is diffusion? A very simple question. So what is diffusion? What is diffusion? You can do that assignment. Yes. So the following questions will help you, and the following questions have been tested in KCSE. Very easy questions. Once you get the concept of diffusion, there is no question you cannot answer. Ensure that anytime you're answering the question on factors, for example, this question number 3, how do the following factors affect the rate of diffusion? How do the following factors affect the rate of diffusion? How do the following factors affect? Thank you. So how do the following factors affect the rate of diffusion? Once you get a question on factors, always make sure that you're giving both sides of that factor. I repeat again, if you're given diffusion gradient, what happens when the diffusion gradient is high and what happens when the diffusion gradient is low? Do not leave your answers hanging. Students, do not leave your answers hanging. So for diffusion gradient, you say when it's high, what happens? When it's low, what happens? Then number B, we have surface area to volume ratio. Then we have temperature. So for each of these factors, give the both sides of the factor. Then these questions can also be asked in form of illustrations. You can be given illustrations, drawn illustrations. We can answer these together, maybe. We can answer these together. So this is an assignment. Ensure you have done it. It will help you a lot and it will help the content sink. So the setup below illustrates a physiological process. So this is the setup. We have this, we have dye molecules represented in black. Then we have a cell membrane. On this side we have water. So dye molecules, dye molecules. Then we have the same setup continuing, ensuring that some of these dye molecules are getting to the other side. Lastly, these dye molecules will become distributed equally on the both sides. They'll become equally distributed on the both sides. So with this illustration, you can be asked, name the physiological process. Name the physiological process. So how do you know which physiological process is being tested? Look at the movement of the molecules. We have them here at the beginning, then at the continuing of the experiment, and at the end of it. So molecules are moving from one region where there are many in large amounts, this, to another region where there are not many. So that simply shows you or makes you understand that the process that is being tested is diffusion. When you're writing diffusion, ensure that you have the correct spelling. Don't leave out the F. We write diffusion with double F. Then a question from the same. Give two examples of processes named above in plants. Which process in plants depend on the physiological process you have named? Which process depend on diffusion? And the question is very specific in plants, not in animals. Please, students, let us read the question again and again and underline the key points. In plants, that is the key point, in plants. So in plants, diffusion aids in gaseous exchange. It also aids in absorption of mineral ions from the soil solution into the root hairs. Then another question, state two ways by which the movement of dye molecules in the setup would be slowed down. State two ways by which movement of dye molecules in the setup. This movement from one region to another, how can it be slowed down? Now this one, they are asking or testing about the factors indirectly. These questions will not always come direct as the ones I've written. Sometimes they can come indirectly. So when you're being asked how you would slow down, how the molecules will now start moving slowly, what can you apply? You are being told to give the factors and also the negative part of the factors. For example, you can lower the temperature, you can increase the thickness of the tissues, you can lower the diffusion gradient. So give your answer according to what is asked. And lastly, this question can be asked as an essay. This question can be asked as an essay where you can be asked, you can also attempt this, to discuss any 6 factors affecting the rate of diffusion. Discuss any 6 factors affecting the rate of diffusion. 12 marks. Stating the factors alone will give you 6 marks. For every correctly explained factor, you get another 6 marks and you get your 12 marks. A very easy topic, a very good topic, and I hope we're going to continue revising. Don't stop revising. After you attempt these questions, don't stop. Continue exposing yourself to more and more questions. I hope you have understood. I am happy to have been here today. Continue working hard, and I wish you all the best. And for now, it's a bye from me.